All right, let's give it a start, guys. It's 8:03. So first of all, for the people online, can you guys see my screen? Can anyone just tell me that you can see the screen? Perfect. Okay, perfect. Well, first of all, I'm actually kind of happy that to see you guys actually face to face, not the camera. And I hate, honestly, I really, really despise it's like old giving lectures online. I think it's the worst thing for me. But I hope that hopefully, we can get everyone here this semester before I leave. Morning. So guys, one thing before we start, the exam. The exam most probably is going to be on the 27th because Eid is Monday's Eid. So basically we cannot have it, so you'll get into the Friday. However, I was basically in talks with the course coordinator, and she was saying technically it seems like it, most probably. So we're gonna if we don't fall into this, we're most likely gonna go into 27th. Exactly. So you never know what happens. So based on that, but also I was speaking yesterday to the course coordinator, and she was thinking that because we're already in some sort of way a bit late with not late, we're not actually that late, thankfully, when it comes to our class, but we've lost what, 3-year class or something. So we don't want to lose a class. So we said to ourselves, well, look, we already have actually the 28th scheduled for exam number 2. That was already scheduled from before. So we'll use that date. So rather than 27th, you have an extra day to study. So it'll be on a Saturday, on the 28th. I still need to confirm 100% with the course coordinator, but this is basically what she was thinking with me yesterday. Most probably. I will definitely confirm with this 100%. I'm just still waiting, basically, with the course coordinator. She's the one who takes those decisions, technically. So once I get to know, I'll definitely send an email as usual. No, Biblos is going to be in person.
And the material is dense, so we'd rather we have it in and the biological students also have organic, and
Also,
they're also trying
to push
they're
it.
trying to push the organic exam, but we're not sure if they're going to push it or not.
No, I get what you mean. Basically, yes, that's what the course coordinator yesterday was telling me. But from what she was telling me is that because those two courses shouldn't be together in some sort of way during the schedule. So that's why she's there and there. She's gonna talk with the people with the chemistry, see what can happen and what not. Exactly. And then they'll decide. Again, the best person to speak to is not me, is actually the course coordinator, because I basically just give the lectures. The course coordinator is the one who gives that kind of stuff. And I'm happy for that, personally. So basically someone's asking, so is the 27th confirmed? Not 100%, not, but I will confirm. I will confirm this week, the date. Did you have anyone had raised their hands? No. Okay, perfect. Okay, so now that's that. This week I'll let everyone know basically when the exam is gonna be. Okay, so now let's go on to chlorophylls and spectra. So as we know, as I said, pigments are the, let's say, molecules that have an amazing, let's say, ability to absorb light. So as I said before, the sun is able to produce literally so much energy, almost about 1 million Hiroshima bombs a day. So, and plants are able to take 1% of that light, 1 or 2%, very little of it. And they're able to do that with basically pigments. So pigments, again, are molecules that are able to absorb light within the visible light spectrum. So visible light, as we said, light, basically, if we go back over here, if you go here, we know that basically kind of wavelength. So basically light can have different wavelengths down to 0.001 nanometers down up to 100 meters. They carry out different energies. The smaller it is, the more dangerous it is. Hence, for example, think about UV light, think about X-rays, gamma rays. These are super cancerous to us. But if you go a bit more later on, radio waves, which were bombarded by the second, they don't do much to us because they don't have that energy to actually, let's say, break macromolecules like DNA or whatnot. So when we talk mostly about pigments, we're talking about the visible light. So this is the visible light we're talking about. And when it comes to the pigments, some pigments basically they have let's say we call this an absorption spectra. So they love to absorb at a certain spectra. So let's give you an example between here. For example, this chlorophyll A. Chlorophyll A loves to basically go up to, let's say, 450, between 400 450, something around that. And then beyond 450 and between 450 and let's say 600, it doesn't. It's not able to do so. So this is what we mean by a spectra. And of course, within plants, or let's say photosynthetic organisms, the main kind of pigments are chlorophyll A, chlorophyll B, and carotenoids. We'll talk about them a bit more in details right now. So, as I said, there's basically these two types. The main pigment is chlorophyll A. Chlorophyll A is one of the only pigments that is able to actually take up this light energy and convert it into chemical energy. What does that mean? It's able to take this light energy and it's able to use it and then convert it to an electron to basically pass on full electrons. That's what I mean when you basically are trying to say that you can convert light to chemical energy. Chlorophyll B, we call this an accessory pigment. It's accessory because it doesn't do this conversion. It's not able to do that. It doesn't have enough power. So it's able to take the light energy and pass it on as light energy, nothing else. This is super important because we use this as an antenna. I will talk about this a bit later on. So if we look basically at the structure of chlorophyll per se, it's this beautiful, literally big molecule. And where? Very simply, you have this porphyrin head over here. And this porphyrin head, have a look at it. It's a big, basically head of just carbon rings, all stuck together. But what you also see is alternative double and single bonds, if you have a look. So just have a look at the double and single bonds. They're just alternating. This porphyrin head is what's responsible for absorbing the electrons. They absorb the light. The light then keeps passing on between the alternative bonds. That's what's happening. It's this porphyrin head that has the ability to do so. And then you have the tail. If you look at the tail, it's hydrocarbons. It's extremely nonpolar. Why would you need this to be nonpolar? Because they're literally situated in a thylakoid membrane. Thylakoid membrane is made of phospholipids. So as we know, any molecule that wants to be inserted in a membrane has to have a nonpolar tail, purely. So that's literally what a chlorophyll is. And if you look at basically the difference between chlorophyll A and B, very, very simplistically, they're just a difference of an R group over here. So if you look at the R group, it's either CHO molecule or a methane, CH3. These R groups are the one that will help the chlorophyll be either an accessory pigment, meaning that they will only pass light energy to light, or they'll have the ability to take this light and convert it into chemical, be it an electron. It's that simple. It's these small little side groups that give you that ability. Okay, so before, when we were literally discovering about, let's say, pigments, we didn't know that pigments worked in a certain, let's say, action spectrum, in a certain spectrum. We had this basically, let's say, scientific thinking. So, morning. So what this means is that we used to think that all wavelengths of light are equally effective in promoting photosynthesis. That's what we thought. We didn't know that pigments actually have a certain light that they prefer. We thought that any light, they could perform it. Okay? We predict if this is true, basically, when we shine light at different wavelengths, we should produce O2 oxygen at the same rate. Because The byproduct of photosynthesis is oxygen. How do you measure the rate of photosynthesis? You measure oxygen. How much oxygen is being produced? Makes sense. All right, let's put this to the test. What they did here is they got some algae. So an algae is basically these plants that you see in the sea. And then they took motile bacteria, so bacteria that are able to move. And they move towards concentrations of high oxygen. This is amazing because then you can literally see where the bacteria is, where the oxygen is going to be. So what you do is you fix, this is basically this massive green algae, this big structure over here. You shine light of different spectrums. So you have basically light here coming from your violet up to your red. So here again, and you shine it. And you see, where's the bacteria going? What you see in this test, basically, if you look at the results, that the bacteria are going to two poles. They don't like to go in the middle. They mostly go to the poles. Okay, this is a bit weird, but this result tells you that there is a certain wavelength that the pigments or the algae likes to basically perform photosynthesis. This was basically what told us that, hey, there is actually an action spectrum. There is a spectra that the most action will occur. And for algae, it's simple. It's in your violet and your far red, because the pigments they have are able to absorb only those. Because, as I said from the beginning of last time, to create photosynthesis, to start it, you need light. Light is the source of energy. You do not have energy, nothing can come up. So we have other accessory pigments like carotenoids. I'm quite sure you've definitely heard of carotenoids. You find these in basically quite abundantly in carrots. Thank you. In carrots. They are super good because they are actually they have a different structure towards chlorophylls. They're more like carbon rings, just carbon rings stuck with a lot of basically alternative single chains. They don't have this big, massive porphyrin head. They don't have it. It's just carbon rings linked to chains. But also carotenoids, they can absorb light at different energies and different basically wavelengths, different chlorophylls. So we technically use them in plants. Plants use them a lot because if we go back, for example, right now towards here, if you look right now, plants have carotenoids, they have chlorophyll A and B. Having these three different pigments allows them to actually absorb light at different frequencies because you have different pigments that are able to actually take different ones. If you only had, for example, chlorophyll A, that means that only at 450, between 400 and 450 we'll be able, and 650 and 700, that's it. But because we have chlorophyll B, we're able to go to 450 to 500. Because we have carotenoids, we can go up to basically beyond 500. We are able to amplify the spectra. It's these pigments that are able to do that. Oops, went too far. But carotenoids, other than that, we use them, plants use them a lot to kind of help them with their action spectra. They have a very good protective role. So, as I said, a lot of these reactions are redox reactions. We're basically breaking down electrons, passing it down. There's always going to be intermediates of electrons being free, of protons being free. These are called free radicals. Their name says it all. They're free radicals. They're radicals that are free, and they're there to basically just be hitting big macromolecules like DNA. These are a big reason we get cancer. These reactions produce a lot of free radicals, and if you are not able to kind of neutralize them, you can get cancer. This is one of their only main diseases, but you can have many different diseases that can come up. So carotenoids are great at neutralizing them. So this is why carrots are very healthy for us. They have a very big, we call them an antioxidant role. When you hear antioxidants, most probably you've definitely heard this, when you basically read any kind of envelopes of these kind of packages you get from foods, they will all tell you these are antioxidants. That means they're able to neutralize these basically radicals. And then you have psychobilioproteins. Psychobilioproteins, different type of pigment. These are pretty good at actually low light frequencies. You see these mostly in plants like algae or bacteria that are basically in the sea. That the light they get is basically really, let's say, very dark in the purple side. So again, because bacteria, these certain bacteria or algae are in an environment where you have a certain wavelength, they basically they have psychobioloproteins. They need to adapt. So this is how they adapt. So this is actually a very nice thing you've definitely seen. So as we know, during spring and summer, we have these trees that have basically these kind of green color. It's very beautiful color green. And then during basically, let's say around October, November, we have this change of color going to more the, let's say, kind of orange-like colors. Why does this happen? Purely, it's the pigments. So what happens during the spring and the summer? There's sun. We have a lot of sun. Chlorophyll A and chlorophyll B are there. They need to start doing all this capture of light. During, let's say, the autumn, we don't have light. Especially people like me living in Finland, literally between November and December, we had 7 minutes of light. That's barely what we had in literally a month and a half. This is why I get depressed there. But what happens during that time, and you really see it extremely there in these kind of countries where you don't have light, these pigments, because they're useless, there's no sunlight to pick up, they die. What happens? Carotenoids come up. Carotenoids have a different color. They basically absorb different colors and they release different colors. So chlorophyll A and B, what happens with them is that when they absorb their spectra, they release the green light. When they die out, you have carotenoids, which are much more abundant now. They release orange light. That's the difference. So this difference in light is not because purely the whole thing is just transforming. It's just that chlorophylls A and B are dying out. And then carotenoids are coming up, so resurfacing, and they're literally shining out the the orange color. So great. Now we know that plants or other photosynthetic organisms, they need pigments. Well, pigments, they're organized together. They're put together multiple molecules in something we call a photosystem. So a photosystem is literally a system that contains all these pigments together. They contain pigments. They contain also basically different types of enzymes in there and proteins. We'll talk about them a bit more. But this basically photosystem is literally on your thylakoid membrane. This is where, as we said, thylakoid membranes in basically your chloroplast are the ones that are taking the light. So within this basically, let's say, photosystem, you have two parts, an antenna complex and a reaction center. Antenna complex is the accessory, basically when you call the accessory pigments. The name is kind of says it everything. It's an antenna. What this antenna does, it literally, think about it as a dish. It's there to literally take up light. It's an antenna complex. It just takes up light. It funnels light. But as I said, these accessory pigments are not able to convert light energy to chemical energy. Great. So they're an antenna. They funnel. They just take all the energy and they direct it towards where they want it. Where do they direct it? To the reaction center. So antenna takes the light in, pushes it into the reaction center. In the reaction center, you have one or more chlorophyll A molecules. Chlorophyll A is the pigment that is able to take this light and then convert it to chemical energy, into electrons. Makes a lot of sense now. You have these chlorophyll B, these carotenoids, that are literally there as an antenna to take in this light. They take it in and they push it. They pass it on because, again, they cannot convert it. They pass it on to chlorophyll A. Chlorophyll A takes it in and is able to push it into chemical energy. That's how it is. Sorry. So the antenna complex, we also call it the light harvesting complex. The name, again, it harvests light. That's all it does. So this is basically how it happens. You have, let's say, a photon, so a light that beams basically into these accessory pigments. So these could be chlorophyll B. These could be basically also carotenoids. They take it in, they keep passing it around until they basically reach finally the center. So again, you have the photon that comes in. It's basically you have these accessory pigments that take in the energy. They funnel it because again, this energy that's being passed is not chemical energy, it's light. Please do not confuse that. It's light energy. Finally, it takes it to basically your reaction center. Your reaction center is able then to transform it to chemical. So that's why you see an electron. So the reaction center takes in basically now this energy, and it takes it now into an electron. So now it converted the light, the photon, into an electron. Perfect. Now you can start doing ATP. Now you can start doing NADPH, the things we need to basically further create glucose. In the reaction center, what you see is the following. This is what actually usually happens in the reaction center. So you basically get light, right? Light comes in, it excites chlorophyll A. Chlorophyll A, basically, now it's excited, its electron it gets energy, and it leaves. So now you've converted this light energy into this electron energy, so this chemical energy. So again, light comes in, it energizes the electron, and this electron now can leave. You have an acceptor, accepts this electron, and of course, now the chlorophyll that lost its electron is oxidized. It lost an electron. Something has to give it back. You want it to basically be recycled back to neutrality. So what you have is you basically have a donor molecule. What that donor molecule is, this could be water, H2O. This could be hydrogen sulfide, H2S. Or this can be coming on from a different, actually, pigment. This will definitely go into what that donor molecule is, because that is very important. But in the case of plants, it's water. This is why we get oxygen. We actually take water, we break water, release electrons, and we literally are trying to do is we're trying to fill this hole that now we've done. So this electron that we lost, basically, over here, we're trying to basically now cover it up. So we lost this electron basically here, we need to fill it in. And that's what water is there for. So when it comes to the light-dependent reactions, these reactions that are only depend on light, we can put them into 4 different categories. First thing is the primary photo event. This is where the pigment captures the photon. It captures it, great. Second is the charge separation. This is where we convert this photon into chemical energy. We excite the electron as much as we can to let it do this photoelectric effect, for that to leave. Electron now leaves, and these electrons actually are transported. Just like we see in respiration, in the basically electron transport system, we're doing the same thing. We're passing now these electrons, and we create ATP to the same thing, chemiosmosis. We have the same complex ATP synthase. We'll talk about this a bit more. So this is how it's done. You take a photon, you convert that photon into electrons, you pass these electrons to create chemiosmosis. We'll go more details about these. So there's 2 types that can happen here. So you see sometimes, specifically in sulfur bacteria, they have just one photosystem, purely one photosystem. That's it. These, basically, sulfur bacteria, they do anaxogenic photosynthesis, which basically means they do not produce oxygen. Why do they not produce oxygen? It's purely because they only have one photosystem. And they only produce ATP. They do not produce NADPH. we'll speak about this specifically, why this happens, because it's very important for you guys to know. So this is literally the photosystem that they have. They have one. We call it P, so photosystem 870. Why 870? Purely because that's the wavelength they absorb. That's it. The name is as simple as that. Photosystem 870, because it's the photosystem that absorbs at 870 wavelength. So what happens is you have a photon that comes in, and this photon it's going to be taken by your antenna complex, so your accessory pigments. They'll pass it on until finally they reach your reaction center. They keep passing it on and they reach the reaction center. Great. Your reaction center now has the energy, the light energy, and it converts it into chemical. Electron. Electron is gone. And now basically it removes the electron and it passes on to an electron acceptor. Again, we're repeating the same thing as we see in oxidative phosphorylation and this electron transport system in your respiration. We're passing electrons. So you have an electron acceptor, which then passes it on to this BC1 complex. If you guys remember, we saw this BC1 complex before. This BC1 complex is actually in cellular respiration. This BC1 complex is a proton pump. It's going to keep giving out hydrogens out. Is basically going to push, and you're going to create, again, a proton gradient. By creating a proton gradient, it's going to go down back into ATP synthase. And you're going to produce ATP. Here, this electron has to come back. Because again, remember, this now reaction center that basically lost its electron is oxidized. It's positive. We need it to get back to neutrality. So the electron does literally a whole circle. And it goes back to its basically original molecule. This is why we call it cyclic. It's a cycle. This does not give you enough energy to produce NADPH, sorry. This is why cyclic only gives you ATP. You have enough energy, this electron, that gives you basically ATP. That's it. So purple non-sulfur bacteria do not produce NADPH. They only produce ATP. When it comes to plants, when it comes to others that actually produce NADPH, they have two photosystems. So we call them non-cyclic because they're not cyclic. They don't come back into again. These have two photosystems. Photosystem 1, we call it P700, sorry. P700, again, it's a photosystem that absorbs light at 700 nanometers. And we have photosystem 2, P680. P680, again, photosystem 2 that basically takes light at wavelength 680. Those two are the ones that are able to create both NADPH and ATP. So this is what happens. Oh yeah, by the way, the evidence between this. What really helped us, let's say scientists, discover that there's two photosystems? Well, what they did is they took a plant and they shined basically two different types of light. One that is the far red. This is the 700 nanometers light. And then they basically when they shine far red light, you can see that the rate of photosynthesis increases. So you see, it goes up a bit. Now when it goes off, nothing. No more oxygen produced. When they turn on red light, this is 680 nanometers, again, you get to see a bit of photosynthesis. But the interesting part they saw is that when you turn on both, you had synergy. Suddenly, you're creating much more oxygen. Why is that? Why is that you're shining one light at 680 and another light at basically 700, that you're getting double, even more than double of photosynthesis? This was the evidence that there is two photosystems, that they're working together. One is taken at 680 and the other one is taken at 700. This was in 1950s that people started to realize that there's two different photosystems. So I'm gonna go straight into here. So this is just basically all of this is just a bit of notes there, but I want to go straight to the diagram because it's much more easy to describe it. So what happens is you have photons that are coming both into photosystem 2 and photosystem 1. So you see, they come in here and here. Let's now focus on photosystem 2. Okay? In photosystem 2, what happens? It takes basically the light. You have, again, what you have is these antenna complexes, the accessory pigments, taking it up until they pass it on technically towards your reaction center. Your reaction center here in photosystem 2 has two chlorophyll A molecules, two of them. They take this light and they're able to release two electrons because they're two chlorophyll molecules. One chlorophyll A molecule gives one electron. So to give two, you need two. They pass it on to plastoquinone. Plastoquinone is another basically electron acceptor. Remember these quinones? We've heard these quinones again. These are in aerobic respiration. We see them in the electron transport system. There are electron acceptors. They accept electrons and they pass it on. They will pass it on to this B6 complex. B6 complex, again, is very similar to the BC6 complex. It's a hydrogen pump. It pumps out hydrogens. This will give you now the gradient for basically of literally hydrogens to do chemiosmosis. So photosystem 2 is able to produce ATP. That's amazing. So the B6 complex gets the electrons, passes it on to plastocyanin, and plastocyanin then basically passes these electrons to photosystem 1. So again, I'm going to repeat this again, photosystem 2. Photosystem 2 takes in the photons, passes it to the antennas, reaching the reaction center. Reaction center gets now excited. It has two chlorophyll A molecules, loses two electrons. Awesome. They pass it on to plastoquinone. Plastoquinone passes it on to basically B6F complex. The B6F complex is a beautiful hydrogen pump. It produces hydrogens out to create a gradient, to produce chemiosmosis, to produce ATP. Great. It then passes the electrons to plastocyanin. Remember here, this reaction center now is two electrons negative. Sorry, pause. Sorry, it has lost two electrons. So it's two positive because it lost two electrons. We need to give two electrons back. Where do we get them, Ben? Water. Either you get them from water, either you get them from another donor. Could be hydrogen sulfide, H2S, depending what it is. In plants, it's water. You break down water, and water is basically then these two electrons are given to this reaction center. Now this is back to neutral. Great. Photosystem 2, done its job, and back to neutrality. Photosystem 1, very, very similar. It gets a photon, it gets excited, it has two basically chlorophyll A molecules, release two electrons. These two electrons are then passed to ferredoxin, which then are basically, ferredoxin gives it to NAD, P+. So what happens is that ferredoxin passes these electrons to NADP+, with the help of NADP reductase, it's an enzyme, you get NADPH. So you have the formation of NADPH. So photosystem 1 is what's helping us actually producing NADPH. This is why we do not get NADPH in the cyclic, because we only have one photosystem that's only able to give us basically ATP. Having two photosystems allow us to give us both. Remember now this reaction center in photosystem 1 lost two electrons. We need to give it back. Well, it's given back by plastocyanin from photosystem 2. So this is where we see these two photosystems working together. Photosystem 2, it helps photosystem 1 go back to neutrality. Photosystem 2 gets its electrons from water, it's done. Photosystem 1 takes it back from the photosystem 2. Does this make sense? Do you want me to go over this again? I am more than happy to do so. Okay. So if you look now basically at the photosystems, photosystem 2, if you look at it, it's very similar to what you see in the purple bacteria. It does the same job, right? In the cyclic. They do the same job. They do ATP. They are very similar to each other, actually. They actually are made of 10 transmembrane proteins, these are the ones that are doing the electron transport systems. So these are the ones that are passing the plastoquinone, these ones. There's 10 of them, basically. And they're basically made of two chlorophyll molecules. That basically they're called they're two P680 chlorophyll molecules. The two, basically, they're chlorophyll A molecules that absorb at 680. The difference between this photosystem 2 and the one you see with the bacteria, the ones that basically do not actually produce NADPH, is very simple. It's actually 4 manganese atoms. So photosystem 2, this one over here, has 4 manganese atoms. So four magnesium atoms. This one here does not have it. So this one here does not have it. The one in the cyclic, the one in the bacteria. That difference of 4 magnesium atoms is what helps photosystem 2 break water. This is why photosystem 2 can break water compared to to one in bacteria. It's these 4 magnesium atoms that are able to do that job. And of course, within the photosystem 2, we have this B6F complex. This is, again, a proton pump. The proton pump that when it takes electrons and it passes electrons out, it gets energy to pump out basically protons, basically hydrogen protons outside. And they create this beautiful, let's say, gradient, which then get back passed to ATP synthase. Photosystem 1, this is the one we do not see in the bacteria. This is the one that is able to do NADPH. It basically is made of 12 to 14 proteins. Again, these electron transport proteins. And they also have 2 chlorophyll A molecules. We call them P700 because they basically absorb at 700. So this photosystem 1 that removes its electrons, it accepts electrons from photosystem 2. So what photosystem 1, its main purpose is to create NADPH. So it passes electrons that now it lost to produce NADPH. That's what it does. So again, this is a very nice, let's say, overall summary of a picture. So what happens? You get a photon. So this is your photosystem 1. Sorry, photosystem 2, sorry, here. This is photosystem 2. It takes a photon, great. It's energized. It takes the antenna complex, takes this energy, this light energy, it passes it on to the reaction center, specifically to the 2 chlorophyll A molecules. It loses 2 electrons. It loses them, it passes them to plastoquinone, which then passes them on to B6F complex, which then passes it to plastocyanin, and then basically this electrons helps the B6 complex to basically get energy to pump out hydrogens. Keep pumping out hydrogens now into your thiaclide space. Great. Now in your thiaclide space, you're creating so much hydrogens in there that the outside, the stroma, is so negative and has very little hydrogens. So through passive diffusion, they're going to go back. We have, again, what we see in cellular respiration, ATP synthase. Again, this big, massive rotor that produces ATP. So it's an ion channel, the F0 complex. It takes inside the hydrogens, and then you have this rotary pump. This basically literally knob is going to keep going on in circles to produce ATP. It's the same thing we see in cellular respiration. Nothing really changed here. This photosystem 1 over here now basically takes a photon, it loses 2 electrons, which passes on to ferredoxin. Ferredoxin then basically passes its electrons to NADP plus, and with the help of the enzyme NADPH reductase, you catalyze this reaction. You basically create NADP plus hydrogen to NADPH. That's what it is, as simple. And now, this basically photosystem 1 that lost 2 electrons, it basically goes back to neutral through the electrons coming from photosystem 2, specifically from plus to sign in. Now, photosystem 2, that lost 2 electrons, will gain its electrons from breaking water. So it's magnesium atoms, it's 4 magnesium atoms, break water and take the electrons. That's literally it. So again, in this process, very similar to what you see in cellular respiration, chemiosmosis, we're using a proton, basically gradient, to funnel energy, to give us energy to produce ATP. Same thing. So in non-cyclic, this is super important for us to know, please. Non-cyclic, basically photosynthesis, you produce both NADPH and ATP because you have both photosystems. Photosystem 2 produces ATP. Photosystem 1 produces NADPH. In the cyclic, you only produce ATP. It's a search circuit. So non-cyclic phosphorylation really helps us to actually build organic molecules. And this is what we're going to be teaching. I'll be teaching for you guys basically on Wednesday. So here I'm going to stop, basically. So just 1 second. What? Yeah, we have good, perfect. You have 6 minutes. Okay. So please, guys, as you can see, it's a big process, but it's a quite simple process. A very elegant, similar to what you see with electron transport system. Very similar. It's just the annoying thing that you need to know now the names. That's mostly the differences here. But is there anything you guys want me to go over, repeat? More than happy to do so. Anyone online? Anything? Oh, if there's nothing, guys, have a nice, enjoyable continuation of your day. Again,
Doctor,
yes, tell me.
I have a question.
Tell me.
Um, is we have chapter 6 and 7 for the exam?
Yes, chapter 6 and 7, yes.
Okay, thank you.
No worries. All right, guys, enjoy your day. And again, if you have any questions, please do let me know. Have a great one. Thank you, guys.